Done, sir.
Okay, so let's start with this session, which would represent the best that this committee number 8, can offer. Our first speaker is Professor Sudha Bhattacharya, who is a senior scientist at Ashoka University, to which he moved after a long stint at the School of Environmental Sciences, JNU. Her undergraduate degree was in botany, and she did her PhD also from an agricultural university. After her postdoctoral work in some of the best universities in the USA, she moved back to India in 1986 and started the first molecular parasitology lab in India. She worked extensively on the genomic organization of the human parasite Entomobia histolica. And in 2005, in a multi-institutional project, the genome of the organism was published in Nature. She is a fellow of all the 3 science academies, and she has shifted a little bit from her interest, and her research focus is now on genetic disorders. She is the co-founder and trustee of World Without GAD Myopathy, a non-profit organization set up to promote research towards understanding and treating rare genetic disorders in India. With this, let me invite Professor Bhattacharya to deliver her talk.
Thank you very much, Piyush. Sorry, Pinak. I'll share my slides.
Yes.
Can you see my slides?
Not yet.
Not yet?
No. Can't see the slides.
Yes.
Can you see it now?
Yes.
Okay.
Do the full screen. And
Full screen.
yes.
Yeah.
Done. Yes.
Is it okay now?
Yes, it's fine.
Okay, fine. So thanks a lot, Pinak, and thank you for inviting me to give this talk. So as Pinak mentioned, I have now started working on GNE myopathy, which is a rare genetic disorder. And like thousands of other rare genetic disorders, for GNE myopathy also, there is no treatment available. Since it's a genetic disorder, so the most direct way would be to introduce a copy of the normal gene into the patient, and work in that direction is going on. But there's also another way that instead of introducing the gene, one could introduce messenger RNA, which could then copy the normal protein and supply the deficient protein in the cell or in the body. So that's what I will be talking about today, that how can we use RNA for therapeutics. So, so far in the pharma industry, the mainstay has been the small molecules. And that is also for very good reason, because these have really served us good purpose. But the problem is that only 10 to 14% of proteins in mammals or in humans are actually druggable targets for small molecules. So the vast majority of proteins are not druggable. So small molecules have limitation. Similarly, recombinant proteins which are being used as drugs, they also have their own limitations because they need to be properly folded with the correct post-translational modifications to be active, and the cost of production is very high. So with the limitations that are there in the available drugs, it leaves open a vast space for nucleic acid-based therapies to move in. And that's how DNA and RNA have now moved into the therapeutic space. So just going back a little bit historically, now to think about RNA as a therapeutic molecule, we have to first see that how did life begin for RNA in the minds of biologists. So the first time that we thought about RNA was actually based on the central dogma of molecular biology. It was more like a messenger, that is to carry the message of DNA into protein. So that's how RNA actually began its life in our minds, that it was basically its purpose was to get translated into proteins. Subsequently, when Jacob and Monod's classic work on the lactose operon, when they showed that actually genes can be regulated in bacteria, and they can be turned on and off depending on environmental cues. So it so happened that here also the molecule which was regulating, although they did mention in the paper that it could as well be an RNA, but it turned out that the repressor molecule was actually a protein. So it was generally thought that genes are regulated by DNA binding proteins. And all the subsequent operons that were studied also showed that it was proteins which were doing the job. So it was quite firmly in our mind that RNA basically plays a role of protein translation. But the observations began to emerge, like in 1981, Tomizawa's lab showed that RNA could actually have regulatory roles as well. So when the copy number of this CoL1 plasmid increases beyond a certain amount, then there's an RNA molecule that actually sequesters away another RNA by complementary base pairing. And this RNA, the first RNA, was actually needed to provide a 3' hydroxyl for replication. And if it's sequestered away, then replication stops. So this regulatory role was assigned to an RNA. And after that, hundreds of RNA molecules were shown in bacteria to have various regulatory roles. They could act in cis like riboswitches or in trans to do a variety of regulatory roles. However, it took the work of Fire and Mello to show double-stranded RNA can be a trigger for gene silencing, which was the first time that it actually captured the imagination of a large number of biologists that RNA could actually be such a major regulatory molecule. And that's because double-stranded RNA, the way it was shown, could function at so many different levels. So the double-stranded RNA molecule was processed, and then it could either act at the level of DNA and silence the gene, or it could act at the level of RNA. And depending on whether it was an siRNA or an miRNA, it could actually cut the RNA and inactivate it, degrade it, or it could spare the RNA, but it could just inhibit the translation. So it opened up a really whole new world, so to say, in which RNA had this vast multitude of regulatory roles. So one can say that turn of this century has seen, in a way, the RNA revolution. And that has also contributed to the use of RNA in therapeutics. So now we have RNA therapeutics in various forms. We have, of course, messenger RNA. We have the antisense oligos and the small RNAs, siRNA and miRNA. We have RNA aptamers. We have short activating RNAs, which actually can activate transcription from a gene. And we have the guide RNAs like used in the CRISPR-Cas system. So let's look at a little bit more about these systems. Now, before we go into details of each RNA therapeutic modality, let's look at some of the key developments which led to the successful use of RNA as a drug. So, of course, starting from the discovery of mRNA itself, some of the technical developments like in vitro translation of mRNA, in vitro transcription using the phage RNA polymerases, which were commercially available, and this commercial kits which were available made in vitro transcription really very doable job in any lab. And then delivery of RNA into cells and tissues through liposomes, lipofectin commercially available. All these developments helped advance the thought that RNA could be used for therapy. But the first major breakthrough paper that came was in 1990, where it was shown that you could actually inject naked messenger RNA of, in this study, they used reporter genes and they injected it into the mouse muscle, and these reporter genes were translated. So that was really great that you could put RNA from outside into the mouse and it gets translated. But in this paper, they didn't show whether it had a phenotypic effect and whether any phenotype was also changed because they used reporter genes. So in 92, another paper came out where they actually showed phenotypic effect by injecting vasopressin mRNA in rats, and they could show transient reversal of diabetes insipidus. So these two papers kind of set the stage for the use of RNA in therapeutics. But there were major issues that were roadblocks. And after these two papers came out, actually the field kind of stopped because the major issues were like RNA being very rapidly degraded by ribonucleases, which are very abundant in the tissues, and the poor uptake because of its negative charge, and the strong immunogenicity. That was a major hurdle, that it was highly immunogenic. Of course, this strong immunogenicity later on became also a blessing to some extent because it was used in mRNA vaccines as an adjuvant effect. So now, so when all these hurdles were there and the field had kind of stalled, we must say that the person who did not give up was this scientist called Catalin Kariko. Currently, she is a vice president of BioNTech. She was in the University of Pennsylvania working in this area. And for more than 10 years, every experiment of her was failing because she could not get mRNA to translate sufficiently in tissues. Finally, in 2005, she got the major breakthrough paper that was here, where she showed that she could suppress the immune activation of RNA by using nucleosides which are modified nucleosides. So basically, RNA, as I said, stimulates the immune system, and that is a problem that is very deeply associated with RNA therapy. And this stimulation is through toll-like receptors mainly, and through other receptors also. So this was known, and so Kariko reasoned that why the innate immune system gets activated by RNA could be actually a defense mechanism which mammals have evolved against bacteria. And the reason is, so how the innate immune system recognizes bacterial RNA and differentiates it from its own RNA is because we know that bacterial RNA is much less modified compared to mammalian RNAs. So about 10 to 25 times less modified nucleotides are there in bacterial RNAs. So what Kariko did was to make in vitro transcribed RNA in which she included modified nucleotides. Many of them she tried. And what she found is that if she used this RNA with modified nucleotides, then, and these are all naturally occurring modified nucleotides, So she showed that RNA containing the natural nucleoside modifications was far less immunogenic and had greater stability and greater translational capacity. So that was a big breakthrough which she showed in 2005 and which then started moving the field forward. In 2008, she then showed that the best modified nucleotide was actually pseudouridine. And so now pseudouridine is used much more often when we are using in vitro transcribed RNA for therapeutic purpose. So it had superior properties. It was not immunogenic, and it was more stable. So it increased the translational capacity also. And further, she showed that actually even after you make pseudouridine incorporated RNA, you still get some immune reactions. And that was because of double-stranded RNA contaminants in the preparation. So if you purify by HPLC and remove the double-stranded RNA, then that preparation is much better. So with all these observations, finally now we find that pseudo-U containing messenger RNA is really the best bet and a good tool for using RNA for in vitro and in vivo applications. And now every part of RNA, of messenger RNA is actually optimized. So including the 5' cap, the 5' UTR, ORF 3' UTR, and the poly A tail. Every part of it has been optimized to get the best expression. And expression, of course, is a function of both the half-life of the RNA and the translation capacity. So when you have optimized both and you have stable RNA and at the same time optimally translated, then you find the best results. But as you can see here that even there, actually the amount of product, the translated product doesn't last for too long and it's gone in a few days. So that is one problem with RNA therapy, that the RNA is not very stable. And so you need repeated dosing. You can't just give one dose. But at the same time, it also actually becomes a boon in a way that the toxic effects are less because the product doesn't stay for too long. And if there is a toxicity, it can be reversed by playing with the dosing. So now let's look at the different classes of RNAs which are used for therapies. So we have the RNA aptamers, which can bind to their particular target molecules and modify their activity. Then we have the small RNAs like the antisense oligos. Then the siRNA and mRNA, which can all hydrogen bond with their complementary base targets. And they have different effects. So the effects could range from destruction of the RNA to alternative splicing to translation inhibition. And of course, we have mRNA. So looking briefly at each class, the antisense oligonucleotides, these are the short DNA or RNA oligos complementary to a target region in their RNA. So the use of these oligonucleotides was actually pioneered as early as the early 1980s. However, again, here the problem was the stability and the affinity for their target RNA and immune activation, and also the ability to induce RNase H digestion, which is needed for the ASO to show its effect. So these were problems that had to be overcome. And here, what they did was to use chemical modifications. These are not modifications which are naturally occurring. So a range of modifications have been tried, and these have actually provided stability to the oligos, and also they have reduced the immune activation. However, there is a problem of toxicity. So now combination of these modifications are used to optimize each application. These ASOs, they can work in two different ways. Some of them are RNase H dependent. So when the ASO binds to its target site, then it is cleaved by RNase-H, and it results in the knockdown of gene expression. And there could also be RNase-H independent ASOs which block, they don't cut the RNA, but they block translation or they induce alternative splicing. Now, ASOs have been very successful so far, and there are a number of ASO drugs that have received FDA approval. So I am not going to list all of them, but just as an example. So here is a drug which has been used for a genetic disorder called spinal muscular atrophy. And it works by including an exon, which is otherwise excluded. And inclusion of this exon restores the activity of the protein. Then there's exon dis51 is for Duchenne muscular dystrophy. Here, there's a premature stop codon and in the exon. So the ASO promotes the alternative splicing, and that removes the exon with the stop codon and restores the reading frame. Another one, Texady, this one is for a disease called familial amyloid polyneuropathy, another genetic disorder, and is caused by mutation in the transthyretin gene. So here what happens is that the mutation results in an abnormal protein, which is toxic. And when that accumulates, then that causes the disease. So the antisense oligo hybridizes to this mRNA, the TTR mRNA, and inhibits translation. So here the phenotype is restored because the translation inhibition of the toxic product is abolished. So this gene TTR actually has been the target of many therapies, which I will be describing now. And the reason is because here you need to actually stop the expression of the gene, and it's much easier to stop expression by the available RNA drugs than to restore expression. So this gene has been used by a number uh, of companies, which is of course good because it also helps to develop the technology. However, it is limited to only stopping the expression. So the small interfering RNA, the siRNAs, they are generally because they hydrogen bond completely with their target, and then after that the target is cleaved. So they are generally used to knock down the target gene. Excuse me. Several siRNA-based drugs have also received FDA approval. So both the drugs are actually by the same company, Alnylam. And here again, these drugs, they inhibit the translation of the target gene. In both the cases, different genes have been targeted and it inhibits the translation of the target gene. mRNAs, now mRNAs go or like siRNAs, only that each mRNA can target multiple mRNAs. So therefore, it can have more than one effect. And it is processed in a two-step process. The first step is in the nucleus by the Drosha enzyme, and then it comes into the cytoplasm and cleaved further by Dicer. And finally, the mRNA, which is formed in the Argonaute complex, unlike siRNA, it does not fully hydrogen bond with its target. So mRNA drugs can be basically of two types. mRNA mimics are the ones which mimic an already existing mRNA and they can downregulate target genes. And mRNA inhibitors, they can inhibit mRNA which is there in the tissue, and that way they can upregulate the target gene. So both of these mimics and inhibitors are under trial, but so far none has been approved. They are under clinical trials, and they are mainly being tried for cancer. Then aptamers are short single-stranded nucleic acids, which are selected by an enrichment process, and they can bind to the targets and modulate the activity. So one aptamer-based drug has been approved, and this is against VEGF, and it is used to treat macular degeneration, but many others are in clinical trials. Now, mRNA drugs, mRNA can be used as a drug in various ways. So the first one is replacement therapy, which is what we look forward to in genetic disorders, because the mRNA can now replace the gene, the protein which was missing, and it can replace with the normal protein. So here, several are in trial. None of them has yet been approved. And Moderna and BioNTech have now become very popular household names. So Moderna has several products in the pipeline for various genetic disorders. And Translate Bio also has for cystic fibrosis, they're testing one molecule. The other application of mRNA drugs, of course, is in vaccination, where with COVID-19, we all know that how successful it is. And then it is used for cell therapy, where the mRNA is actually transfected ex vivo into cells of the patient. And then these cells have an altered phenotype. And when they're given back to the patient, then they can help in overcoming the disease phenotype. So, but apart from the COVID-19 vaccines, none of the mRNA-based drugs have yet approved, but there are many of them are in clinical trials. In the vaccination approach of mRNA, there's a straightforward approach where you just introduce the gene which codes for the antigen, and then the antigen is expressed and elicits the immune response. But interestingly, there's also another method which is being tried, that is the amplification of mRNA, where they have actually put replicase gene of a viral coded replicate RNA-dependent RNA polymerase genes, which actually amplify the RNA, so that now the amount of antigen produced is higher, and it can give a more successful immune response. In cell therapy, of course, is being tried a lot for cancer immunotherapies, where the patient's cells are taken out and then transfected with the mRNA of the cancer antigen and then put back into the patient. The other big push is now for the CRISPR-Cas technology for gene editing. Here also the work is done by the RNA. So CRISPR-Cas is an adaptive immune system of bacteria where they can sense the invading RNA of plasmid or phage. And in an RNA-based, the invading DNA of the plasmid or phage, and this DNA is then differentiated from the cell's own DNA by the use of small RNAs which guide or nuclease to the target that has to be cut. And then the target, which is the invading DNA, is cut by using the guide, which is a small RNA. So this has been cleverly now used so that you can make the guide RNA from a gene that you want to disrupt, and you use a Cas9 system to cut the gene and edit it specifically at the place that you have in mind. So obviously it is a technology that can be used for gene editing in genetic disorders. And it is being applied in various cases. So all of these CRISPR-based therapies which are in the pipeline, they all work by disrupting the target. So far, that's how they are working. So we would hope that in the future, it will also be that they can introduce the correct nucleotide in place of the defective mutant nucleotide. That so far has not been accomplished, but the disruption by using Cas9 has been used. So, for example, here in edit 101, there's a mutation in the intron of the gene, which causes a splicing defect. So this intron is then cut, and then it rejoins by non-homologous end joining. And so that results in actually normal expression of the gene. The splicing defect is removed. Similarly, in this other application, CTX001 is for blood disorders like sickle cell disease and beta thalassemia, where there's an enhancer which is disrupted by the Cas9, and that promotes the expression of fetal hemoglobin. So fetal hemoglobin can then, to some extent, do the job of the missing adult hemoglobin. Then there are these others which are by Intellia Therapeutics. Intellia Therapeutics is a company that has been promoted by Jennifer Doudna herself, and they are using various targets, again the TTR gene in liver and kallikrein gene for hereditary angioedema. Now, one major problem with RNA-based therapies is the delivery. How do you deliver the RNA to the tissue or the cell? So the large molecular weight of the RNA and its negative charge, it is a hindrance. And the cellular barriers have to be overcome, not only the cell membrane, but also the endosomal escape, which is a major problem because a lot of the RNA is actually lost in the endosome itself. So there are various modalities that are being tried. For local administration, like in tissues like eye or brain or tumors, where you know exactly where to inject the RNA, there people are trying even with naked RNA and they are getting some success. But targeted delivery is being tried by conjugating the RNA with, say, an antibody. Conjugation with GalNAc has worked very well for targeting to liver, because liver has this GalNAc binds to the acylglycoprotein receptor in liver cells. And so GalNAc is working very efficiently to target different RNA molecules to the liver. Cationic polymers are also being used for delivery of siRNA. And there's an interesting concept here which is being developed, that is a self-immolative materials which can lose their cationic charge, and that enhances the endosomal escape. So this is being developed. And of course, lipid nanoparticles are very much in use as they are larger in size and they can accommodate a large amount of RNA. So lipid nanoparticles have been used for the siRNA drug Bhattacharya, and they are being modified in various ways. And now in this particular case, they have included cholesterol and PEG. So cholesterol provides stability to the LNP, and PEG enhances the circulation time so that the LNP stays in circulation for longer time. But then it can also have a negative problem that if the LNP doesn't get cleared and stays in circulation for too long, then it could also have some toxicity. Then the surface of LNP is also being modified to help in targeting to specific tissues. So mRNA has some very important benefits over DNA, for example, that in the case of mRNA, it functions in the cytoplasm itself, so it doesn't have to enter the nucleus. Whereas for DNA, it becomes even more inefficient because it has to enter the nucleus in order to express. So mRNA, we don't have a problem of insertional mutagenesis because it stays in the cytoplasm and there's no entry in the nucleus and no genomic integration. Now, mRNA is transiently active and it degraded. So although this is a problem because you have to keep on administering the dose repeatedly, but it is also a boon because the toxicity effects can be controlled and you can control the dosing. Whereas with DNA, it is more of a one-time shot, and you have to understand the dosing, etc., right in the beginning so that you give the correct dose. Otherwise, you cannot control it later on. mRNA is also simpler to produce. It is less expensive. And since it is based on in vitro transcription, so you avoid the use of cells for making the RNA, and that reduces the toxicity. In DNA, because we so far use cell cultures, so there, and the plasmid backbone itself could have some issues. So therefore, this is a problem. But a new option is now being developed called doggy bone DNA, where you use a rolling circle mechanism to actually produce large amount of DNA in the test tube. So this could help now improve the kind of DNA that is being administered. So I told you about all the various forms of that RNA therapeutics has taken, but there are still a lot of challenges that have to be addressed in mRNA therapy. The major challenge is the translational efficiency of the mRNA. Now, we still don't have enough protein being produced from the mRNA, which is transfected into cells and tissues. The amount that is being made is okay for vaccines, because there even nanogram to microgram amount of antigen is sufficient. But when you want to treat a genetic disorder or for other kind of applications, for cancer also, you may need to have much more quantity of the active enzyme or antibodies, which could be in milligram or even gram amounts. So to jack up the efficiency to that level is a challenge. Then the delivery to different organs, so far liver has been, because liver is by itself an easier organ to target, since naturally molecules tend to go to the liver. So efficiency of delivery to other organs apart from liver, that remains a challenge. Then the internalization of RNA into the cytoplasm and endosomal escape also has to be more optimized. So whereas all these challenges are there, there is a huge number of efforts which are going on in this direction. And so it is quite, one can be hopeful that in the years to come, all these challenges will be overcome. So the newest kid on the block right now is the use of circular RNA, or this company called Laron has named it endless RNA. So this RNA is circular, and since it is circular, so it is much more stable, resistant to exonucleases, and it is also not recognized that much by the immune system, so it has less of immune activation. And what they have done is to introduce the internal ribosome entry sites so that this can now be translated. And what they claim is that the duration of protein expression from this RNA is quite long, because it is resistant and it stays much longer in the tissues as compared with linear RNA. And what they say is that this could actually keep getting translated in the tissues for even months. And that's a very big plus, because with linear RNA, it's only days or weeks. And after that, you have to administer the second dose. So here, if they can push it to a few months, that will be really a big leap forward. So to conclude, in the last decade, we have seen RNA therapeutics has moved very rapidly. Earlier, it was only a dream, but now it looks like a reality. And I have only named some of the major companies, but a lot of biotech startups are moving in the direction of RNA therapeutics. Possibly, they have been encouraged by the success of COVID-19 mRNA vaccines. So that is not to say that there are no challenges. A lot of challenges do remain, but for those patients of incurable genetic diseases, RNA therapeutics seems to be holding a real hope. Thank you.
Thank you, Sudha. for a very nice exposition of RNA therapy, how the problems associated with it and how the impediments can be overcome, the delivery modalities and benefits of mRNA over DNA therapy. So many of us have learned very many new things. And let me see if there are questions or anybody present can ask question. I don't see anything in the chat box. Maybe I will start by asking a question. You said you mentioned of TTR gene and the mutation causes some disease. Now, when you use the oligonucleotide, do you target the original gene or the mutated gene? Has it to be specific for the mutation?
Yeah, it has to be specific for the mutation because you're going to target the mutated gene.
Yes.
And the mutation was causing a splicing defect. So now when you target the mutation, then it is the mutated gene that will now be able to express.
Yeah, okay. Any other question? Yes.
Sudha, this is Madhu Dikshit. Thank you so much for very, very illuminating talk. I was just wondering that while we are looking for the safety studies on these candidate molecules, are there any specific type of safety which is required to be done?
Specific type of safety features, it's not really anything specific to RNA, but as we know that, some of these oligonucleotides, especially which use unnatural modifications, they can cause toxic effects. So the toxic effects differ from application to application and what modification is being used. So the problem is that, even if you test the toxicity in mouse, but it may not be toxic in mouse, but it may be toxic in humans. So it needs repeated testing in various animal models to be sure
Okay.
that there's no toxicity.
Okay,
Yeah,
thank you so much.
there's no specific issue. Yeah.
Mhm.
Okay, and it seems that there are no questions at the YouTube site also. So I think it's time to thank Professor Bhattacharya for a wonderful talk, and we'll move to the next talk. Thank you, Sudha.
Thank you. Thank you so much.
Oh, do we have to log in again or this one continues, no?
Yes, sir, this will continue.
Okay, because I got some this thing join meeting. So this one is continuing. Okay.
Yes, sir, yes.
So, but I don't see anything. Okay. Sankar, are you ready to start? Then I will introduce you.
Yeah, I can. Yeah. So I'm here.
Okay.
I'm here. Yeah. So ask Sudha to unshare so it becomes easier.
Okay. Okay. Actually, I am stuck somewhere. Yeah, I think Sudha has to come out of.
I think she has to unshare. Then
Yes, Sudha, you have to unshare and come out.
I'm just doing it.
Yeah, yeah. Okay.
Ma'am, you don't have to sign out.
Have you unshared, Sudha? Or
Not yet.
not yet? Yeah.
They have to go, to the bottom. Maybe somebody can help. The INSA people are, I think, there. Ask them to go to the bottom and then
There will be an unshare button, red button that will be
at the
for
end.
unsharing
at
the
the bottom.
slides.
Yeah.
You can just click on that.
Hello, can you hear us?
Yeah,
Yeah.
trying to.
So at the bottom of this, WebEx thing,
She
you
has
will see
lost. She has lost the full screen display. I don't know what is happening. WebEx I don't use, so I'm not very.
No, the bottom of that WebEx panel, there will be an unshare thing. Wherever she's shared, there will be an unshare.
Full screen is not coming, so that's what it is happening. We are not able to get the full screen on.
You can click on this join meeting, that green button. You can just click on that, then it will come.
Oh boy.
You can remove her from that.
No, the stop share button, you can get it while going up towards the top.
No, it is actually she is not getting her full screen coming up.
Join meeting.
So that is the problem. That's why.
Ma'am, I will just remove you, then you can join again from the same link.
Yeah. That will be better. That will be better.
Yeah.
Okay. Yes, sir, she has left. We can continue.
Okay.
Can you see my slide?
I can see your slide, but yeah, I can't read anything from my computer. I thought I'll take a few points from your profile, but anyway, I all of you know very well. So let's move to the second talk. Dr. Rajan Sankaranarayanan, he is from CCMB. His background is physics, so he's supposed to be intelligent like all physicists are, but then he moved to do his PhD in molecular biophysics unit at IISc. He is one of the finest structural biologists in India, working among other topics, proofreading in protein biosynthesis and how chirality is recognized during this process. I think he will be talking on that. He is fellow of all the 3 science academies in India. Among his many awards and laurels, some notables and recent ones are Shantiswarup Bhatnagar Award, G.N. Ramachandran Gold Medal, and Infosys Science Prize last year. He is an associate editor of Journal of Structural Biology, and since he publishes routinely in eLife, I find that he is also in the board of reviewing editor in eLife. With this, let me request Sankar to deliver his talk.
Sure. Yeah. So thank you, Vinak, for that kind introduction. And you people able to see my slide? If anyone can tell me, that will be great. Then I can move on.
No, it was visible, but now it is gone.
You are not able to see it?
No, but it was there when I started.
Okay, so let me try to. I hope she has already unshared it, correct?
It is stop share, sir. You can share it again.
I'm not able to see my PowerPoint now. Okay, hold on.
Have you opened your slide on the desktop?
I've just opened now. Okay, so
Yeah,
are you able
it
to
has come.
see this now?
It has come.
I'm going to the slide mode.
Yes.
One of you can confirm.
Yes,
Are you
you are
Perfect,
set.
sir.
You are
Okay,
all
great,
set.
great. Thank you, Pinak, so much, and INSA for this opportunity. Pinak mentioned that since you come from physics, that you associate some intelligence. The only intelligence that I can associate is that
You are mute.
oh. Where do I?
No,
No,
sir, we can hear.
No, I can hear you.
Yeah, we
yeah.
can hear.
So is it fine now? Can you still hear and see my slide?
Slide
Your
went
slides
out.
are
No,
gone.
but now we see you.
Oh man. Okay, I'm going to do this again. I'm sorry. Something. Is it fine now?
Yeah, absolutely fine.
Okay, great. So the only intelligence that you can see, even in terms of operating this online platform, one can associate with me is that, like some of our colleagues, I moved to biology, I think, at the correct time. So what I'm going to talk to you people today is about the work that has been going on in our lab on a process called proofreading, which is happening during the translation of the genetic code. So the last lecture, Professor Sudha Bhattacharya talked about different versions of the RNAs and their role in translation. That translation means is in terms of using a research work or a research output for multiple purposes, for clinical purposes, for human use, or any other biotechnological applications. However, the molecule that I am going to talk about is another version of the RNA, which is one of the first RNA to be characterized very thoroughly and also identified, which is the transfer RNA and its role in translation. So the transfer RNA is a molecule which you see over here, and this molecule is the central player in connecting the protein world to the DNA or the nucleic acid world. So each RNA, which is the tRNA represented here, is responsible for charging one amino acid on top of its CCAN, which is depicted here by an enzyme called tRNA synthetase. However, since mistakes happen, they actually have to be proofread. And that is the process that we have been looking at in the lab over the last 20 years. And as we started understanding this process, we ended up looking at.
Shankar, you are mute again.
No, I'm fine. I didn't do anything. Are people able to hear me?
Yes. I think it's fine. I can hear you.
So, Pinak, you let me know if there is any problem at any given point.
I will. You're fine.
Yeah. So, because otherwise it can become problematic. Yeah. Okay, so let me continue. So we actually stumbled upon enzymes that are responsible for removing the opposite chiral entities of L amino acids, which are called D amino acids. And we started looking at them and eventually are finding of late their role, or these chiral proofreading enzymes' role at critical junctions in eukaryotic evolution over the last few years. So that is the direction of work that is going on. So I will just give you a basic information since it's an INSA kind of presentation, which is for a general audience. And then I will move over to some of our recent research. So if you look at, the chiral checkpoints during protein biosynthesis, sorry.
Now I cannot hear you.
You cannot hear me?
No, and your screen is also gone. Screen has come.
No, hold on. There's some problem with all that we are seeing right now.
Yeah, PowerPoint has come. No, it has gone.
Are you seeing this PowerPoint now?
No,
I don't know
yes,
how it is changing.
now it has come. Yes, now it is full screen also.
Okay, so
Hear
I
you.
will not. Yeah. So anyway, great. Sorry, Pinak, for that. So if you look at the accuracy of information transfer, starting from the information content in the DNA to RNA, and then to making of proteins, each of these steps are tightly controlled. And of course, the genomic information is sacrosanct because that is the one which is passed from generation to generation. So therefore, that process is the least error prone. As you can see, the error rates are of the order of 10 to the power minus 8 to 10 to the power minus 10. However, the other processes, which is the translation, which is the process of converting an RNA to protein, these processes are more error prone. And the work that I'm going to describe today is about the error of a particular amino acid when it is attached to a tRNA, because these are the tRNAs that are taken to the ribosome for translation. Now, if you look at the set of amino acids that are shown here, 20 of them which are commonly used by organisms, most amino acids are quite different. Say, for example, glutamic acid, phenylalanine. So the enzymes that choose them have not much difficulty in selecting them with the accuracy that is required for translation. However, there are systems where the difference is only of a single methyl group, like say, for example, isoleucine and valine, and say threonine and serine. So an enzyme which is supposed to be picking up threonine, which is say threonyl tRNA synthetase, would make a mistake once in a while of activating serine. And that error rate is more than what is expected of a faithful translation. And therefore, that error has to be corrected. So now this kind of a problem was first identified even before the protein structural framework were available by none other than Linus Pauling. So Linus Pauling, just by looking at the kind of amino acids and their chemistry, suggested that the biological systems which are going to use them will have problem in discriminating with a very high specificity because they cannot be discriminated very heavily. However, as we know, the observed error rates during protein synthesis are much higher, much lower in the context of these amino acids. So one of the questions which people asked in the field around 80s is, the 1980s, is how these molecules achieve such a great specificity. So in addition to that specificity of related amino acid selection, there is also something that is happening at the level of the ribosome. So if you see the selection at the ribosome, the tRNA molecule that carries an amino acid is checked both for its anticodon, which is the 3 nucleotides, which are paired with the mRNA's codon, and that's how the recognition happens for the amino acid at the ribosomal level. However, nothing is checked at the level of what is attached at the tRNA. So therefore, if a wrong amino acid were to come in here, that is incorporated in the growing polypeptide chain. And now this property of the ribosome is used by many to introduce artificial amino acids or unnatural amino acids in desired positions, which has certain applications. However, what we are interested in here is also the opposite enantiomer. Say, for example, if an opposite enantiomer were to come here, that can be incorporated also in the growing polypeptide chain. And there are recent evidences that suggest that, in fact, the amino acids can be incorporated in the first position. However, the second position, if a D amino acid or an L amino acid were to come, the coupling will become much slower. So that is what people have noted. Now, the editing process works in this manner. I don't know whether this movie would work. So anyway, it's not moving, so I'm not going to disturb this. So you have the CCA end of the tRNA, which takes the amino acid, either the correct or the wrong one. If it is a correct amino acid, it goes towards an editing or proofreading site, which is covalently attached in a single polypeptide chain in majority of the cases, or there can be freestanding modules. So when the CCA end flips, it checks for whether it is a correct amino acid or the wrong amino acid. If it is a correct amino acid, it cannot enter. And if it is a wrong amino acid, it enters the site and ensures that that wrong amino acid is cleaved. So by using this two-step process, enzymes, enforce higher, faithful translation of the genetic code. So when it comes to the context of proteins, as I just mentioned, one needs to look at the opposite enantiomers as well. Okay, so why these proofreading mechanisms are important has come from the work of Paul Schimmel in the early 2000s. What he showed in the case of an enzyme by called alanine tRNA synthetase, even a partial compromise of the editing activity is good enough to create phenotypes like cerebral loss and eventually leading to ataxia. And the reason is that there is going to be now a global mistranslation. So this is not mutational effect of a single amino acid to something else. You are going to have mistranslation of all the proteins that are made in a given cell, which gives rise to this kind of an impact. And therefore, the importance of studying these mechanisms at a molecular level and a mechanistic level came to the fore. In my talk, what I am going to focus is on opposite chiral amino acids. And we all know for protein synthesis, what is majorly used are L amino acids, and the mirror symmetric counterparts are not used for protein synthesis. And way back in the 90s, early 90s, rather, what people have shown is, in principle, you can actually make proteins with D amino acid. And this was the cover of Science, in which Kent and co-workers made a D amino acid HIV protease, which is chemically synthesized, and showed that this D amino acid protease can actually fold, and it's nothing but a mirror image of the L amino acid. It is not just the protein that shifts its enantioselectivity, but it is also the substrate, which is, instead of now acting on L peptide like a L protease, now the D protease would act on D peptides. So both the protein as well as the substrate become the mirror image of the L amino acid counterparts. However, while you can make proteins with full of L amino acids or full of D amino acids, you cannot have heterochiral polypeptide. What I mean by that is, you cannot have substitutions in a L amino acid polypeptide chain once in a while with D amino acids. And this substitution will be even more lethal or disturbing to the folding and functionality of the protein than an amino acid which is shorter or smaller by a single methyl group, and so on and so forth. So the substitution of D amino acids in protein biosynthesis in a cellular scenario has to be avoided at all costs. And therefore, the cell has multiple checkpoints to ensure that the D amino acids don't enter the protein synthesis machinery, which is the ribosome. So the first and foremost is the choice of the amino acid, which is done by the enzyme family called amino acid tRNA synthetases. And as you can see, majority of the tRNA synthetases are highly specific for the L-amino acid counterparts. However, there are quite a few, as shown here, which are not as specific. So therefore, you do have D-amino acids which kind of infiltrate this process. And the second chiral checkpoint in the cell is that of elongation factor. So what people have shown is that while elongation factors are highly selective of L-amino acid tRNA, they also can take the amino acid tRNA with a reduced specificity. So this has been seen. So which means the second step of the process is also porous. And the third checkpoint, of course, is that of the ribosome, wherein the protein synthesis actually happens. Even the ribosome's peptidyl-transferase center has shown to be more specific for coupling L-amino acids than L and D or D and D, suggesting that even there is a kind of a checking which is happening at the peptidyl-transferase center at the ribosome. So therefore, all these three chiral checkpoints ensure that the D amino acids are not put in the growing polypeptide chain, and therefore maintaining homochirality during translation of the genetic code. However, each of these processes leaves some amount of D amino acid tRNA as a byproduct because they are not used. So what happens over a period of time if these D amino acid tRNAs are not recycled to free tRNAs? You do not have enough free tRNAs in the cell, and therefore the translation gets arrested. And therefore, an enzyme which is called D-amino acid tRNA deacylase is invoked in all life forms, which I will show you a little later, to ensure that these D-amino acid tRNAs are recycled to D-amino acid and free tRNA, and therefore they can enter the translation machinery. As I mentioned, if you have excess, D-amino acid tRNA, you can have either global misfolding of cellular proteins, or you can have depletion of the tRNA pool. And therefore, this enzyme called DTD, which ensures that your D-amino acid tRNAs, particularly the D-amino acid, is removed. And the interesting mechanistic aspect of this enzyme, as you can see, is that there is only one D-amino acid tRNA deacylase for all the 19 D-amino acid and 19 tRNAs. And therefore, it has to act with a very high specificity in a milieu of excessive L-amino acid tRNA and ensure that it doesn't attack the L-amino acid tRNA and only attacks the D-amino acid tRNA for its activity. And therefore, the L-amino acid tRNA now can be taken for protein synthesis. So several years back, we solved the crystal structure of the enzyme, which is called DTD, which is present in all bacteria and eukaryotes, particularly it's present across the board. And this is a dimeric enzyme, and we are now zooming it in the active site of the enzyme. And what we found, interestingly, is that when the tRNA is captured with the D-amino acid on top, most of the interactions are happening in one subunit of the molecule. However, the other subunit, which is shown in green here, is responsible for the chiral selectivity. And the first explanation that the structure gave is that this enzyme does not care about the side chain of the amino acid, as can be expected, because this is not a side chain specific enzyme. So the side chain is kept out of the binding pocket. However, what the enzyme checks for is the chiral center, which is over here, which is the C alpha. And when we have a closer look, as we can see here, the gly-cis pro motif. So there's normally peptides are found in a trans-peptide conformation, but the gly-pro motif here, as you can see, is in a cis peptide conformation. And that is responsible for keeping the two carbonyls in such a way that it actually selects the chiral center. And therefore, any L-amino acid, if it has to come inside the binding pocket, will have a severe clash with the rigid gly-cis pro motif, as we observed. So that is how the enzyme selects the D-amino acid. And the next question that we asked as to what is the role of this enzyme in terms of its mechanistic function, which means is it a deselection enzyme or is it a L rejection enzyme? So to test that, we took to glycine, and as you can see here, we could actually solve the structure of this enzyme in complex with gly analogs as well. And also we have done biochemistry to show that this enzyme actually can act on glycine, suggesting that this enzyme operates with a fundamental, mechanistic basis of L-chiral rejection. So anything but L will be allowed to pass through this chiral selectivity filter. And therefore, L-chiral rejection is the modus operandi of the major, chiral checkpoint, which is this DTD1, as we showed some time back. Now, the overall scenario stands like this at this point of time in terms of the structural and biochemical understanding. So, if you look at the cell, you have, L amino acids, D amino acids, glycine. Of course, you have a whole lot of tRNAs. So the amino acid tRNA synthase are majorly responsible for producing L amino acid tRNA, and the DTD is, or the D amino acid removing enzyme, is not able to act on that as expected. So therefore, they are taken for translation. Now, the other extreme is the D amino acid tRNA, which is the mirror image counterpart of L amino acid. And here, the DTD can act whether they are bound by elongation factor or not, they act with them with a very high specificity. One of the questions that we asked when we had the activity on glycine, which is a chiral molecule, is whether this is a mechanistic fault or design flaw of these systems, or that they have any other functional relevance. So what we eventually showed is that the glycine, which is mistakenly attached on tRNA alanine by an enzyme called alanyl tRNA synthetase, is actually the substrate of DTD. Which means to say that the D amino acid tRNA system, tRNA deacylase system, also has another functional role in terms of handling this particular mischarged substrate. However, when it comes to glycine attached to tRNA glycine, we showed that both elongation factor as well as in combination with a particular base called discriminator base in the tRNA, which ensures that the gly-tRNA gly is not acted upon. Okay, so now coming to the next part in terms of how the chiral checkpoints have played possibly a role in eukaryotic evolution at different time points. We look at now the overall picture of DTDs. So as you can see here, in all bacteria and in all, what we call ophistocons, which include both fungi and animals, we have DTD1, and even in plants we have DTD1. So which means all eukaryotes have DTD1. Whereas you have something called DTD2, which is an enzyme which does a very similar job. However, the fold and the evolutionary origin of this DTD2 is distinct than that of DTD1. So therefore, you have two enzyme varieties, one from bacterial origin and the other from the archaeal origin. So we looked at particularly a variant of DTD1 present in what are called these ophistocons, which consists of both yeast and animals. And when we looked at them, they had more or less very similar sequence as that of DTD1. However, in the database at that time, it was annotated as a variant of DTD1, which they called it as DTD2. However, we later named it to ATD, and the reasons will become very clear because it has a totally different function. So when we looked at sequences, what we found was something very interesting. So if you see the GP motif, which is responsible for the chiral selection, as I mentioned earlier, that GP motif is highly invariant in ATDs also. One would assume that it would do a very similar function. However, there are residues that are responsible for, the fixing the GP in a particular orientation. So one of the bright students in my lab identified that maybe they are doing something else because the GP will not be in such a strict orientation. So to understand this, what we did was to solve the crystal structure of this protein. And again, I'm sure this movie is not going to work. So anyway, so let me just go to the next step. So when we solve the crystal structure of this protein at a relatively high resolution, what we found was quite exciting for us. As you can see here, the glycis pro, which in DTDs normally comes to the chiral center, that glycis pro is shifted to a glide trans pro motif. So the GP motif moved from glycis pro to a glide trans pro. And this is the kind of shift that we see because of this. And therefore, the chiral selectivity as expected of DTD was not expected to be there in the case of ATD. And in addition, because of this movement, you also have some space created at that level. Therefore, we kind of predicted that it could possibly be used for activating or acting on some L-amino acid counterparts. What we eventually showed, which I'm not going into the detail because of lack of time, is that while for a long time in the case of proofreading from Linus Pauling's observation that amino acid, confusion, which will have a problem because of which proofreading is required, we showed that when we move from, unicellular organisms to multicellular organisms, and particularly to higher systems, the genome, of course, expanded, because of which you have significant expansion of tRNA. And there is something called the Trna Iso Decoder, which is a common parlance in the tRNA field now. What it means is that unlike iso acceptors, here your anticodon is going to be identical in all these tRNAs. However, you can have variations in the body. So those kind of tRNAs have expanded in the context of animals, and therefore the tRNA ambiguity sets in for the amino acid tRNA synthetase to activate them. So instead of making a mistake with amino acid ambiguity, now you take the correct amino acid, but you will put it possibly on the wrong tRNA. And therefore, you have a problem of amino acid selection, which is correct. However, the tRNA selection, which has gone wrong. And we showed that the ATD is responsible for clearing one of the major mistakes of alanine being charged on top of tRNA threonine. And that is the mistake that is cleared in the context of animals. So what is the physiological role of ATD is the question that we ask next, because it's nice to see that biochemically this protein is working on removing a tRNA misselection error, but what is the cellular role? So we actually knocked out this protein in HEK cells, hoping that something would happen, and to our, whatever, not so lucky scenario in this case particularly, this protein, when we knocked out, nothing happened, which basically means there was no mistranslation, as if that this protein had no real effect in the cell. However, since the mistake is that of alanine or tRNA threonine, we started to look at whether there are other redundant factors in the cell that can possibly play the same role of removing alanine on tRNA threonine. And what we found was, in fact, the threonyl tRNA synthetase, which is supposed to be, removing serine from tRNA threonine when it is mistaken, the same proofreading module can actually remove L-alanine from tRNA threonine also. However, there is zinc which is present in the active site of this editing domain, which is not very tightly bound, making that zinc binding site highly prone for oxidation. And therefore, during oxidative stress, we found that the threonyl tRNA synthetase active site is killed, and therefore now the enzyme cannot act on, and this is where ATD came into the fore. So now we went back to our knockout cells to figure out is ATD essential in the context of oxidative stress. And of course, and that's where we found that when we oxidative stress in the ATD knockout cells compared to the wild type, the ATD knockouts suffered a lot. So I will not describe the details, but these are all recently published work. So basically, not only HEK cells, also ES cells show pronounced specificity. We also did luciferase-based assays in the lab, and what we in CCR, and what we found was that if you have oxidative stress in the ATD knockout cells are highly prone for proteotoxicity, which you see as aggregations. We eventually also showed using mass spectrometry that such substitutions are possible. So then the question that we asked is, okay, here is a protein which has appeared in the context of animals, responsible for correcting a mistake that is happening only at the level of animalia, which is not there across the board. So when did ATD appear in animalia branch of life? And that took us, interestingly, to the set of organisms called kinoflagellates. So what are kinoflagellates? So kinoflagellates are universal colonial organisms. They are the sister group of animals. While they don't have multicellularity as such, but they exhibit multicellular phenomenon in the context of different stress or environmental conditions. And Nicole King from University of California, Berkeley is a pioneer in this area. And what we found is wherever the multicellularity has taken or basically beginning to take its root in terms of bringing in factors responsible for multicellularity, you do have the appearance of ATD at that level. And in not only the ATD, what we also found is that at the level of kinoflagellates is where during a tRNA, what you call tRNA expansion, you have the tRNA error that also sets in, which is responsible for the mistranslation. So you have a direct correlation between the presence of a protein, which is clearing the error, and the appearance of the error at the level of kinoflagellates. And in fact, we went on to eventually identify the protein ATD from this organism, which is S. rosetta, and also showed that biochemically this variant like ATD. So that clearly shows that the origin of ATD is at the level of where the multicellularity is taking root. So to summarize this part of the talk, what we have shown is that there are two things that are happening when you go to animalia. One is a huge genome expansion, which leads to tRNA expansion. And that tRNA expansion gives rise to ambiguity in tRNA selection. And addition, because of this multicellular nature, which is, you have oxidative stress, which is a common thing that happens during this, which is well, studied in the literature. And because of that oxidative stress, one of the proteins that is responsible for clearing such error is becoming inactive. And that is the time where ATD is rooted and eventually responsible for ensuring that this mistake is removed so that the translation can go on, suggesting that ATD is an innovation which is responsible for the emergence of multicellularity. So what we eventually kind of suggested is that such a genomic innovation alleviates mistranslation associated with multicellularity. So we are now looking at why nature has to create a problem at a particular junction of eukaryotic evolution and eventually solve the problem there itself. Is there a way of possibly routing these RNAs for other purposes is something that we are looking at. So I'm now switching gears for the next 5 to 10 minutes, I guess. I hope Pinak will it be all right because I lost some time.
Yeah.
Yeah. Okay.
5 minutes maybe. Yeah.
Okay, fine. Let me try to see if I can hurry up. Okay. So the point, the next aspect that I'm going to describe is another protein which we have also identified as important for possibly land plant evolution. And that is a protein called DTD2. So all along, I have been talking about DTD1 and its variant ATD, which is present in Ophistoponta. So now I'm switching gears to this archaeal version of DTD2. And if you see from archaea, they are not present everywhere else, but they come straight to plants. So basically from archaea to plants, this protein has jumped, and we started looking at what is the role of this protein. Biochemically, of course, it was known earlier, even before we started our work, that this protein is responsible also for removing the amino acids from tRNA, suggesting that every life form should have this activity. Sometime back, in 2004 or so, a Japanese group had shown the role of this protein. So at that time, even this protein was not known to be acting on the amino acid. So in Arabidopsis, this protein was called GEK1. So what they found was that if you knock out DTD2 in the genome-wide screen of Arabidopsis, they found that now the Arabidopsis is highly toxic to ethanol, but the toxicity is not because of ethanol, but the ethanol is converted to acetaldehyde, and acetaldehyde creates this toxicity. So that is what was known in the literature. This is a D-amino acid removing protein, and this protein removes toxicity of acetaldehyde in Arabidopsis. Now, the thing that we asked is, what is the link between the two? What is the role of a D-amino acid tRNA deacylase in removing acetaldehyde toxicity is the question that we asked. And of course, acetaldehyde toxicity is well documented in the literature in the context of animals, particularly in humans, that you have DNA repair enzyme mutations which lead to cancer, particularly in the context of Fanconi anemia mutations, and acetaldehyde can create a serious toxicity for people who have such mutations. So what we hypothesized in terms of identifying the link between DTD2 and acetaldehyde is that possibly there is some modification that is happening on the tRNA because of the acetaldehyde, and that modification DTD2 is relieving. So, but we did not know where the modification is happening. So, long story short, what we did was to look at where this modification happens, and we showed that the modification is happening at the level of the amino acid. So these acetaldehyde can actually make what are called N-ethyl adducts. And since L-amino acid tRNAs are protected by elongation factors, they don't go through this modification. However, the D-amino acids are not as much protected. Therefore, they get modified. And when they get modified, they become very stable compared to the amino acid tRNA, and they remain in the system for long, scavenging the tRNA out of the system. And therefore, the DTD, we wanted to check particularly, is there a crosstalk with somebody? It says, can people mute their speaker? Yeah, please. Yeah. Thank you. So the DTD2, we checked whether it moves that kind of modification, and we showed that eventually DTD2 is responsible for removing this N-ethyl adducts which are attached to tRNA. And we call this activity as a NEDAT deacylase activity or NJ activity, which is very unique to DTD2 and not to any other system. Then naturally the question that comes is, when did plants acquire DTD2? I mean, was it there from the very early lineage of algae, or that it came at a particular point during plant evolution? And when people looked at multiple systems for which genome sequences are now available in the streptophytic algae scenario, you have many important processes, like thyloglucone, plant hormone, and cyclic electron flow, and all these processes have appeared at the level, not just at the level of land plants, but before. Say, for example, many streptophytic algae like Klebsiella medium and Chara have acquired these processes. And each one of them, if you look at now, the plant hormone or cyclic electron flow, they are adapted from bacteria. And some of them have eukaryotic origin. Of course, you do, as can be expected, there are certain plant innovations as well. So in the context of that kind of an analysis, we took all the algae as well as the available sequences of the ancestors of land plants. And very interestingly, what we found was that while the DTD2s are present in archaea, of course, as I mentioned, you don't have them in bacteria, and therefore they are not in the very early branches over of the algase, as you can see from here, but they start appearing from the context of chlorokybus. The chlorokybus is one of the subaerial capsules that you can see, whereas the mesostigma is not. So basically, there is a transfer of this archaeal gene, which is responsible for removing this deamino acyl tRNA adduct, which can come because of anaerobic fermentation. And that is eventually leading to or helping these land plants to basically adapt to a subaerial kind of an environment. And therefore, this is in the context of land plant, evolution genes which are responsible for land plant evolution. In addition to many of these bacterial origin genes and eukaryotic and plant innovations, we identified this protein as of archaeal origin to be, kind of transferred to the land plant ancestors and responsible for possibly land plant evolution. So to summarize this part of the story, if you look at the evolution of land plants, they face two major problems. One of them is the low oxygen in soil, which creates anaerobic stress in roots, which leads to significant amount of acetaldehyde production that is well documented in the plant area. And also the soil, because of the microbial degradation, has significant presence of D-amino acids in them. So you have this problem of D-amino acids and the confluence of D-amino acid problem with that of the acetaldehyde problem that the roots face. You need to eventually have this DTD2 to remove the adducts on tRNA so that proper translation can go on and possibly helped in the, or a key event which helped in the emergence of land plants is what we proposed. I will not have time to go through this story, but another role of chiral checkpoint which we have recently identified in the lab is in the context of mitochondrial emergence, which will be published shortly. So there again, what we see is the role of chiral checkpoint coming to the fore. So what we are seeing again and again as a repeated theme, and which I want to bring it in the context of my concluding remarks, is that the molecules that are there from the very beginning, correct, and with which systems have evolved and which are highly critical. Say, for example, if you look at one of the canonical molecules and one of the first molecules to have possibly originated in the context of life form is the tRNA. And the tRNAs are there from Luca all the way up to whatever the evolution, wherever the evolution takes us. Now these tRNAs are used for different purposes, and the molecule have been fragmented and they are used for regulation. The tRNA as such is used for as a splicing regulator, gene expression regulation, nutrient stress response, and so on and so forth. The amino acid tRNA, or the lack of it, is also used to measure protein turnover, cell wall remodeling, and so on and so forth. Oh. Hello?
Okay.
Yeah. So therefore, the molecules which are present in the system for a very, very long time are used again and again for different functions, and that too at critical junctions. And the same is true for amino acid tRNA synthetases. As you can see, they are involved in plethora of functions. So the canonical molecules that are present for a very long time in a system have been used for other functions as well. So what I would like to conclude my talk is with, since these chiral proofreading molecules, as we have identified, they are involved in critical stages of eukaryotic evolution, like say multicellularity, or in the evolution of land plants, or in the context of mitochondrial. And this is strengthening a kind of view that if I have a fold in a biomolecule in a system, or a fold, that fold, when it is very rigid, it is used many, many times, the molecules are decorated by many, many changes, and they are used for different functions. And therefore, is there a possible code which kind of tells the system that which molecule to go for? And maybe that is a theme which we are seeing repeatedly, but we generally think that it is because of chance. It's just a chance event because this molecule is available and therefore this molecule is used. That's the current thinking. But how, when we see these themes recurring, more often, is there a code which is hidden and the code is being used at some point? And where will we go for finding the code and what could be the code is a direction one can possibly look at. So with that, I conclude my talk by thanking all the members of the lab. The work that I presented today is the PhD work of students Santosh Kumar, Mazid, as well as Jotin, who is currently in the lab. They are assisted by Raghavendra Singh, a postdoc, and Bhatnakar, who's another postdoc, in a more significant way, with help from others. And I thank you all for your attention. Thank you.
Okay. Thank you, Sankar, for a wonderful talk, which shows how, based on starting from structural studies, how one can ask many interesting biological questions that span across defined organisms, bacteria, animal, plant, how diseases can manifest, and the evolution of molecules to perform diverse function. It's a very nice talk. And let's see if there are questions in the chat box. Not really. Anything at the YouTube side? Let me start by asking, since there is no question here, a very hypothetical question which you haven't addressed and you are not supposed to address. Now, you have looked into the chirality at the alpha carbon atom. One amino acid, threonine, it has a chiral center in the side chain.
Sure.
Now, supposing we have a very artificial system where we put in a lot of threonine with the chirality which is not present in nature. Do you think there will be, both the chiral centers are present for threonine side chain. Do you think your system will be able to discriminate between two? As far as I can see, it is only looking at the C alpha, that center.
Sure. So you have answered it, Pinak. So basically, that is the center, which is the C alpha chiral center is the one that is looked at. And the other side, correct? Any extension of that will go outside the pocket. And there is no recognition of that chiral center at all. So in principle, if you have a L, correct, threonine with, a chirality which is accepted normally, correct? Which is the L chirality at the C alpha, followed by a different chirality, at the second position, you will be able to, charge this, correct? And this enzyme will not be able to recognize that as a problem. Did I answer your question?
Yes. Yeah, that's what I also felt.
Yeah, because the selection is mainly centered around C alpha.
Yes.
And
that could be the reason why some of the amino acid tRNAs like alanine, it can also bind glycine. That's what you said, because glycine can fit in there.
Absolutely. Because chirality rejection is mainly for L chirality. So you cannot have a C alpha and connected to a C beta, which is in the L form. As long as you have a glycine which is having only a hydrogen, that can be accommodated in that pocket.
Yeah. Okay.
I think Sudha has her hand raised. So
Yeah. Hi, Shankar. Very interesting talk, as usual. Congratulations. So I just was linking your work with, my now recent thing of expressing RNA in tissues. So I just wanted to know how much would chiral proofreading affect the translation ability of mRNA, and whether it could be boosted by, let's say, upregulating these enzymes, or it's not really, it doesn't really have much effect on efficiency of translation.
Yeah, these are responsible in a normal system to be present to take care of these, exigent scenarios. So I don't think by overexpressing them, actually, what happens is if you overexpress, which you have shown in bacteria, that the normal level of expression does not affect the glycine attached to tRNA glycine, because as I mentioned, this enzyme can also act on glycine, correct? So if you overexpress these enzymes, then it can deplete the cellular glytRNA pool, and therefore it can create growth toxicity for bacteria. We have shown, and we have also seen some indications in the context of eukaryotes. So the levels of these proteins are also critical because they cross-react to the amino acid. So one needs to be controlling them. So overexpression as such may not be a great strategy to boost whatever cellular fitness or whatever that we are looking at.
Okay, that's very nice to know. Thank you.
Yeah. Okay.
Any other question? Or not really. Okay. Is there any question? No. Okay. If not, let's thank Sankar for a wonderful talk, and also both the speakers in the session for keeping us enthralled for last 1.5 hours. Thank you all. Yeah.
Thank you.
Thank you.
Thank you. Bye.
Bye bye.
Any message from INSA or not? I think nothing really. Huh?
Thank you, sir.
Yeah. Okay. It's done. Okay. Thank you all.